Okay, so we're just going to dive right into it. So we're going to start, and we're just going to review really quickly the central dogma. Um, this should definitely be a review. I mean, I hope at this point you guys are familiar with it, but we're definitely going to go over it because you really should know it for the test. So DNA, RNA, protein. So it starts with DNA. Eukaryotic DNA is double-stranded. It's a molecule that does not leave the nucleus. It's very important, so it stays sequestered in the nucleus. Um, in order to shuttle this information to the ribosomes for translation of proteins in the cytoplasm and the rough ER, because that's where our ribosomes are located, enzymes in the nucleus create a single-stranded RNA molecule in a process that's known as transcription. And that undergoes further modifications and it eventually makes its way to the ribosome. So the ribosomes then translate the mRNA's nucleic acid sequence into an amino acid sequence to create a polypeptide, and that process is known as translation. So that was just a quick review of the central dogma. It goes from DNA to RNA to protein. So going from DNA to RNA is known as transcription. Going from RNA to protein is known as translation. And we're going to talk a little bit about those two. So from nucleic acids to amino acids, the genetic code is the unambiguous code that ribosomes use to translate 4 nucleotides into 20 amino acids. So if you look over to the right, this is known as the genetic code. There are 64 combinations. 61 of them code for actual amino acids, and then 3 of them are what is known as a stop codon. So the 4 nucleotides that we find here and throughout DNA are adenine, guanine, thymine, and cytosine. So in RNA, the nucleotides are actually adenine, guanine, cytosine, and uracil. Thymine is not present in RNA. It's uracil that actually pairs with adenine. And it's important to note that it pairs during transcription, but RNA is a single-stranded molecule, whereas DNA is double-stranded, so DNA is always paired. RNA, the pairing is really used for replication. The pairing is used for transcription. During translation, words containing 3 nucleotides known as codons are used to decide which amino acid is to be added to the elongating polypeptide. So like I said before, there are 64 possible combinations. A lot of times, if the first 2 of the 3 letters are the same, it will remain the same. The amino acid that is produced will remain the same. That's not always the case, but that's just something that I wanted to throw out there. That's usually something useful to know. And then 3 of them are stop codons, which means once that sequence, once that codon is registered, it's sort of like a termination signal for the for transcription. I'm sorry, translation. So like I was saying, there are 64 possible codons. 61 of them code for amino acids, and 3 of them are known as stop codons. And we also call them nonsense codons because they don't code. They're not actually coding. They're just there to send a signal to stop creating that polypeptide. So different codons that code for the same amino acid are known as synonyms. So if we take a look at this code, say we look at this first box all the way up to the top left, you have UUU and UUC. Both of those are coding for phenylalanine, so they would be considered synonyms. So because a specific codon will always code for the same amino acid, the genetic code is said to have no ambiguity. It's unambiguous, which is a pretty important concept also to understand. So if we look at phenylalanine again, for example, there are 2 different codons that will code for phenylalanine. However, each one of those codons will only ever code for phenylalanine. So we can have multiple codons that lead to the same amino acid, but that singular codon is always going to code for the same amino acid every single time. So that's why we say that the code is said to be unambiguous. It doesn't have any ambiguity. And then there are some exceptions to the central dogma. So for example, retroviruses utilize an enzyme known as reverse transcriptase to go against the dogma and create DNA from RNA. So like I mentioned before, this is the 15-week supplement course. So a lot of the stuff that we're talking about here, we did go over the original course. We do have a 15-week MCAT prep course. It's for free. You can find it on YouTube. You can find it on our website. So I did speak in one of the virology lessons about reverse transcriptase and the lysogenic cycle. So that's the most common example of how certain organisms or certain viruses can actually defy the central dogma. Okay, so this brings us to the first question. Just take about a minute. It's a pretty simple question. I just want to make sure that everybody is comfortable reading that code and that chart. So which of the following codons will no longer produce the same amino acid after its third letter has been changed from C to A? So pause the video, take a minute, and we will go over it. Okay, which of the following codons will no longer produce the same amino acid after its third letter has been changed from C to A? So the correct answer is A, and that would be UAC. So it really just comes down to looking over here. You just have to look and see which codon is producing a change. It's not going to produce the same amino acid. So UAC, if we change that UAC to UAA, let me find that UAC. If we change that C right over here in this top third box to an A, it's no longer coding for tyrosine, it's becoming a stop codon. So that mutation would actually be heard, that wouldn't be silent. So that's why UAC is the correct answer. And B, C, and D are incorrect just because if we're changing the C to the A on B, C, and D, these answer choices, they're going to code for the same amino acid. So we'll just look at one of them, and then if you guys would like, you can definitely go back and look at yourself. But CUC, so we have our first letter, C, second letter is U, so then that brings us right over here. So if we change the C to an A, it's still coding for leucine, so there really hasn't been a change, and that is the case with all of these answers except for A.